Here's my video for the fundamentals of chromatography, which is the 9th lab for Chem 1412. For this lab, it was using provided food dyes to perform chromatography experiments in order to interrogate the components of the dyes used in various Kool-Aid colors and flavors, and then this could also be used to look at items in the home for their different components and physical properties that could be found using chromatography. Okay, so I'm beginning the fundamentals of chromatography lab, and I'm going to weigh 2 grams of each of the Kool-Aids and put them into each of these cups. Okay, here I have my 2 grams of orange Kool-Aid, strawberry Kool-Aid, and grape Kool-Aid added with 5 milliliters of purified water. Okay, I am now going to weigh 0.1 grams of sodium chloride to 100 milliliters of bottled water to make my 100 milliliters of a 0.1% sodium chloride solution. Okay, so here I have prepared my 0.1% sodium chloride solution. I have dissolved the salt and I am ready to move on. Right, so I went ahead and cut the chromatography strips. I measured 1.5 centimeters up and then cut points in the middle and then measured 9.4 higher than that bottom line to draw the top line of the chromatography. Okay, and here I've prepared the micro tips of the pipettes by cutting off 5 centimeters of the ends, and I'm going to discard the bowl. Okay, so I'm beginning activity 1 for the known F, D, and C dyes. So I have my 3 chromatography strips and my 2 drops of each food coloring solution placed onto my plastic bag. Okay, so here I have spotted my 3 chromatography strips with my food dye, and I have added 20 drops of my 1% sodium chloride solution to each of my 3 test tubes. And I am now going to continue. So I've inserted the straight pins into my chromatography strips, and I'm going to enter them into the test tubes to perform the experiment. So here I have my 3 chromatography strips from activity 1 with the blue number 1 here, yellow number 5 here, red 40 here. So this is where the solvent came up, and I will be marking the center of the dots. Okay, so moving on to activity 2, I have my orange, strawberry, and grape Kool-Aid solutions, and I'm going to go ahead and spot these 3 chromatography strips with the microtip pipettes, and I have the solvent prepared here, 20 drops of that in each test tube, so I will be placing those in and seeing the distance traveled to calculate the RF values for that. Okay, so here I have each strip spotted and pinned, so it's ready to go into the solvent. Okay, so here I have my 3 chromatography strips for the Kool-Aid. This one's grape, this one's orange, and this one is strawberry. So you can see the variances in dyes. So I'm going to calculate those and match them with their dyes. Okay, so here's just a little summary video of activity 1 and activity 2. Here I have my chromatography strips from activity 1 with the different dyes on them. I've calculated their RF values. And here are my chromatography strips for activity number 2. For activity 3, I dissolved some of the orange sprinkles into a little bit of water, purified water, and spotted a chromatography strip. So I'm going to add that to the solvent and see what happens. So this one's really faint, but there's this light yellow right about in here, and the red hardly moved. But it appears that the yellow and the red are what was in the orange sprinkles. So I added my third trial of the orange sprinkles chromatography strip to my overall sheet, and the color wasn't as concentrated as the other trials, so it's a little bit harder to see, but I will add that to my data as well.